Hello, and this is the webinar for UCSC Xena and TOIL data. My name is Mary Goldman. I'm at the University of California, Santa Cruz at the Genomics Institute here. We just started right at 11, so I'm actually going to wait just a couple of minutes for just to make sure everybody is able to see everything and gets it from the beginning. So in the meantime, I'm just going to start in just a few minutes here. I just want to remind people that if we're using Google Hangouts on the air, this is actually our first time. We've done a couple test runs, but this is our first time really doing it to the public. So if you have any questions or things are not working quite right, feel free to send me an email. I do have access to my email still while I'm doing this. So feel free to send emails there. If you want to ask questions during the webinar, there's the Q&A app. And so I will be able to see questions there. Feel free to post them at any point in time, and I'll be going back and checking through them periodically. So we're going to give people just a few more minutes, and then we will start. Excellent. I see our first question here, which is so great. That makes me so happy. So I think what will be best is if I give the presentation first, and then answer questions there. So although I'll just give you a little preview, is the download the bulk raw normalized data sets. So we do have an S3 bucket, an Amazon bucket that has that data. And so once you have access to that, but I'm not gonna, I can send a link out at the end to everybody who signed up, and I'll include the link to the S3 bucket. All right, so why don't we get started here? So like I said, my name is Mary Goldman. I'm at the University of California, Santa Cruz Genomics Institute, and this is the Xena TOIL webinar. So actually, before we jump in, I'm going to really quickly just do the acknowledgements, just because they always end up in the end and you never see them. So we work with quite a few people. We have a small team here and a lot of different collaborators. So thank you to everybody who helps make this possible. So an overview of how this webinar is going to work is we're going to cover what is Xena, and then we're going to talk just really briefly about what is TOIL, so you understand what the TOIL data is. We're going to do an example in the slides, then we're going to do a demo, and then there'll be time for question and answer at the end. And we purposely made this webinar on the shorter side. When I've given this before, it's taken somewhere between 30, 35, 40 minutes, kind of depends on what kind of questions people ask, but should be fairly short, so we'll have lots of time at the end for people to ask questions about anything that they want. So let's start out with what is Xena. So Xena was conceived sort of looking for seeing that there's this real big need in cancer genomics for a shared standardized global network of genomic data. So you're never going to have all the genomic data of the entire world in just one place. There's just too much data that's being generated at too high of a rate. And so there's not going to be just one location that you go to get your genomic data. You're going to be getting data from a lot of different places. Some of these repositories are going to be quite large. Maybe they're sponsored by the government, like NCI, or these can be also small studies as well, you know, people who are just doing a little small sequencing projects within their own lab. So Xena was sort of conceived of thinking about this. So I'm going to go over the architecture of Xena, and there's really two parts of Xena. The first part is this federated data hubs. So like we were saying, there's all these different places you're going to be getting data, and so there's these data hubs at each of these locations. It's a distributed system to host cancer genomics data and the clinical phenotype data. Right now, the data that we host is really quite varied. Mutation data, SNPs, indels, even large structural variants, copy number data, expression, methylation, and then all the different clinical and phenotype data that you can think of, such as age, tumor subtype, anything like that. So the federated data hubs come in two flavors. The first flavor is a private data hub. This is what we're talking about. We're talking about small labs doing some sequencing. So these are designed to be really easy to install on your laptop. It installs much like any other program that you download and install. So, you know, it has a little wizard that takes you through installing. You can load data into it either using the application, or if you have a lot of data, you can also load it up by command line. We support Mac, Windows, and Linux. And if you, you can install it just on your laptop, or you can also install it behind a firewall. Maybe if you have some data that you want to share with within a lab, but it's protected, you can put it behind a firewall, and you can also restrict access to the data hub. So the second flavor of the private data hub, or I'm sorry, of the data hubs, is the public data hubs. So these are very similar to the private data hubs, except they're public and they're in the cloud. So I'm going to go over some of the large public that we have here. And I'm just going to go through, just in case not everybody's familiar with the data, I'm going to talk really quickly about what these different data sets are. So the first one is TCGA, probably one of the more popular ones. Has 20,000 adult tumors. And what makes this data set really powerful is that you're looking very longitudinally, or sorry, not longitudinally, but it's a really comprehensive view of that tumor. So it has the copy number data, it has the expression data, it has the mutation data, it has the protein expression data. So it's a really deep view into individual tumor samples. Similar to that is ICGC. And so ICGC is pretty much like TCGA, except it's an international group rather than just the United States. Then we have the PCOG data, and this is 2,800 whole genome tumors. So this is looking, rather than going across a lot of different tumors, this is instead looking really deeply, looking at things like the promoter region and the enhancer region, looking at areas outside of the you know, the quote expressed part of the genome. Then we have the target data. This is looking at, again, it's very similar to TCGA, but in this case it's for pediatric cancer, so fewer samples, but still very comprehensive and very useful. And then the last one, so the ones we've been talking about so far are all cancer genomes. The last one is actually different in that it's benign samples. So in this case, it's 10,000 benign samples. And what GTEx is doing is it's looking at the expression data for these benign samples. So you have sort of a baseline or an idea of what normal expression might be, quote, normal expression. And then you can compare that to a tumor sample. So what we did right there with the data hubs, both the private and the public, and that's one component of the Xena system. The second part is the Xena browser. So the Xena browser allows you to connect to all these different data hubs and view the data together in one place. This happens dynamically from your web browser. each of these data separately or together, integrated or not. It's very flexible. Right now, we support Chrome, Firefox, and Safari. So just a kind of a overview of what the system looks like in a more picture-like way. I'm going to actually turn on, I think I have a laser pointer here. Yeah. So you can see here all these different public data hubs here. They're coming together in one place on the browser here. And you can see here they're also connecting to a private data hub. And notice that none of the data in here is going to the cloud and then coming back down. So this data is staying secure. As secure as your laptop is how secure that data hub is. And then you can also connect to data from an institutional Xena hub. And these lock icons show that you can restrict access if you would like. All right, I'm going to turn off the laser pointer here. So that was an overview of what Xena is. And now we're going to talk about TOIL. And I'm not going to spend as much time about TOIL. Just really a very brief overview. So TOIL is a pipeline architecture that was made as part of BD2K and GA4GH. This is all at Santa Cruz. And GA4GH, just for people who may not know, is the Global Alliance for Genomics and Health. Basically, it's a architecture for computing large amounts of data, and they have designed it in such a way that it's very fast and it's very cheap. And so as a proof of this architecture that they've put together, they uniformly reanalyze data from TCGA, TARGET, and GTEx. And this was really great because previously the data from TCGA and the data from TARGET and the data from GTEx, they all had RNAseq expression data, but it was all analyzed using different pipelines. And so these pipelines can have computational batch effects. And so TOIL went in and analyzed it all together underneath the same pipeline, removing those computational batch effects, which means that you can now compare TCGA data to GTEx data. And so Xena is the host for this data. We offer it up for visualization, analysis, and download. All right, so now really quickly check to see if there are any questions. Looks like there's not any questions here except for the question we're going to get to at the very end. I still see it there. Excellent. All right. So for the example, I'm just going to give you a little seeing ahead, I guess you would say. Both the example and the demo are going to be the same. So in the example, we're going to kind of go through the biological background behind it, give you a little bit of an overview of what's going to happen. And then in the demo, we're going to go bit by bit through the example in live on the web. And you're welcome to follow along with that. So for the example, we're going to be looking at FOXM1 and its different isoforms. FOXM1 is a transcription factor involved in cell cycle progression. So we're going to be looking at 3 isoforms of FOXM1. The first one is FOXM1A, which is actually a transcriptional repressor. And then we're going to be looking at FOXM1B and FOXM1C. And these are transcriptional activators. And if we look here, I'm going to turn on the laser pointer again. We can see FOXM1C and FOXM1B undergo exon skipping here. And so you can see that these two, even though there's the gene, that these different isoforms have actually quite different effects. This one being a repressor and this one an activator. So we're going to look at the TOIL transcript level data to dynamically view the data across these 3 different data sets here. So when we start out here, the default view that Xena drops you into is called the visual spreadsheet. So in this case, each row is a single sample like a spreadsheet and similar to something like Excel. So you can see this is one sample here, or I'm sorry, these are the samples here along the side, and along the top we have our different columns of data. So right now we just have one column of data, which is the cohort. So this is showing you where the data is coming from. So in this case, data is coming from TCGA, Target, and from GTEx. And it's important to note that the data is sorted left from right by any column of data that you might choose. So we're going to have column of data here, column of data here, column of data here, and it's all going to be sorted by this first column here, and then subsorted by columns thereafter. So now we've added FOXM1 expression to this view. Remember, one sample is the same all the way across here. And what we can see is that FOXM1 expression tends to be higher in TCGA and target tumors. So we can see here for the expression data, red is relatively overexpressed and green is relatively underexpressed. And so we can see here it tends to be lower in the normal samples that are coming from GTEx and relatively higher in the tumor samples coming from both target and TCGA. But what's interesting is that if we not at the whole gene, but instead at the different isoforms, we can see that FOXM1b and FOXM1c, which if you remember are the activators, are overexpressed in the tumors compared to the GTEx normal samples up here, and that we actually don't see that same pattern in FOXM1a, which if you remember is the repressor. And then if we look FOXM1 regulated genes, we just 4 genes that are one. And you can see a really strong pattern between the isoform expression here of these two isoforms and the regulated genes here. So that is just the example. Oh yes, and then just really emphasizing that you're looking at a lot of samples here, almost 20,000. And so I'm bringing this up both to, it's just quite a lot of data to be looking at all at once, so it gives you a better feel of what you're actually looking at here. But also as a note that during the demo, because we are looking at such huge numbers of samples, it will take a second or two for each of the different columns to load up. So just a little reminder to be patient for that. So I think right now we're going to be going on to the demo. I am going to stop presenting the screen, and then, oh, excellent, great. I'm glad we're checking in here. Can the TOIL pipeline be used for private datasets in Xena browsers? So right now the TOIL pipeline is published. Oh, here, I can actually select it. So now it says I'm answering perfect. So the TOIL pipeline, can it be used for private data sets in the Xena browsers? So in this case, the TOIL pipeline is published, so you can run it yourself. There's also, I know that it's been run on AWS. Right now, because it does cost money to do that and we're not funded to compute it, but you can compute it yourself, and we can point you to the people who did the TOIL pipeline and help you set up there. We've also had other people just compare their RNA-seq data using the same RNA-seq caller, what is Callisto. Great, thank you. These are great questions. And then I'm going to take this next one here as well. So this one says TCGA data has normal samples as well. How should we take care of this during comparisons? So that's a really great question, and something that I think is actually still kind of open in the field. So one thing I'll say is that the TCGA data, the normal samples, they actually, not very many of the normal samples have RNA-seq data or have RNA-seq analysis that was performed on them. It's actually a pretty small subset. So in general, it's not there's just not a lot of data there, which might reduce your statistical test, the power of your statistical test, just because of how small it is. There's also been a really big a push lately within the RNA-seq community, saying that the matched normals are often, perhaps they are not as good of a quote normal as we might, as the tumor microenvironment is quite large and is affecting all of that tissue. So even though they're picking a sample that looks normal, it still might be affected by the tumor microenvironment, and it may not have quote normal expression. So a lot of people have been pushing for the GTEx data, which is, there's no tumor there at all. However, that still really depends on your analysis and your point of view on this debate. So it's sort of an ongoing debate. I've heard compelling arguments on both sides. So at this point, I would say it's really up to you. The only caveat I would say exactly is what I said before, the TCGA data, the normal samples, there's just not much RNA-seq data. So for power purposes alone, you might choose to use the GTEx data. Great. These are really great questions. All right. So now I am going to choose. Great. So we're on the demo portion now. So at this point, people should feel free to follow along with me here. So we have, this is our home page, UCSC Xena. So it's at xena.ucsc.edu. You can see, I'm just going to point at the link, has a little 5-minute tutorial on some of our most used features. So that's a really great resource. We're actually going to scroll down here to the Xena public data hubs, because TOIL is on one of our public data hubs. We're going to click Learn More. So you can see here's some public data hubs that TCGA, just like I all said before. We're going to scroll down here and click on the TOIL data here. So that is going to take us to the TOIL landing page. It has a little bit more about the pipeline if you're interested in that. It also has a preprint. So if you're going to use this data, we ask that you cite this preprint here. I'm going to scroll down to the bottom. That shows you the 5 different cohorts, the 5 different groups of samples that we have here. We have the GTEx data. We have the target data. And then we have a group where we put both the TCGA and the target samples together. We have just the TCGA, and then we have TCGA, target, and GTEx all together. And so we are going to start out with this group here. So, because we want to view all 3 of those data sets together. So I'm going to click on Visualize here. This will take us to our main application. It'll take just a little bit to start loading up the samples. As you can see, the samples here along the side, this is our visual spreadsheet. And then we're going to have columns of data along the top, and we're going to add data here. And again, note, so actually the data has been updated, so there's even more now. There's about 19,000 samples here. So we're going to click on the plus data button. And the first column of data we're going to add is that cohort. So that's in the phenotype data. Click here, and then you're going to click Next. Then we're going to click on Study, is this. So you can see here the little spinny wheels telling you it's thinking about it. It's going back and collecting all of that data for 19,000 samples here, and then is going to sort it and put it on the screen for us. So we can see here, this is the study. If we tooltip over, we can see individual samples here. I'm actually gonna use I'm gonna grab and drag here, make it a little bit bigger and a little bit skinnier actually, just to make it so that we can fit more data on the screen here. So now we're going to look at FOXM1 expression. So we're going to go here. We're going to go to gene expression. We're going to add the RSM data here. And we're just going to put in our gene here, FOXM1. And then we click done. And again, it's going to think about it a little bit just because it's pulling 19,000 samples here, which is quite a bit. And I'm sure actually if anybody's following along, they're also hitting the server as well. So we can see here exactly what we saw. We're just remaking that figure, relatively lower expression in the normal, relatively higher expression in TCGA. And again, I'm just going to make this a little bit smaller. Now for the next one, so this was FOXM1, we're looking at the gene. Now we want to look at the transcripts. So if we click the plus data button again, now we're going to look at transcripts. We can look at the TPM data, RSM. So we're not going to be entering genes. We're going to be entering identifiers. And we can see here the identifiers are ensemble transcript IDs. Sorry, ensemble transcript IDs. So we're going to need to enter those. So if you are following along, this is a great place to go back to your email that I gave you and make copy and paste those ensemble transcript IDs because I certainly don't have them memorized myself. And you're going to click done. And again, it's going to build out that data. It's going to take us there longer for this one because we're pulling 3 different transcripts all at once. And again, it's for 19,000 samples, so that's just pulling a lot of data over. So we can see exactly what we saw before, these different transcripts. And we can see here individual expression for individual samples here. One thing that's nice is that you can change the labels here. So those transcript expression, or I'm sorry, those transcript IDs don't mean much to me. So I'm going to put in these more human readable terms for myself. So that way I remember what these are. All right. And then last, we're going to add those regulated genes. So we're going to go to the gene expression data for this. And then again, I'm going to go and copy that data and then put it in here. These are the 4 genes. And then I'm going to click done. And then while that's loading, I am going to go back to Google Hangouts, make sure that there's no more questions. Excellent. Hopefully people are able to be along with this. Feel free. You can also say, slow down as a question. It's a good way to get hold of me here. And I'm going to really quickly. So, yep, here we can see these different genes, relatively higher expression corresponding with this isoform expression here. So now we have done what was in the example. I'm going to give just a couple more, show a couple more features of Xena. So I wanted to show people that you can click and drag to change the sort order. So this is really helpful. Like I was saying, each sample is the same all the way across, and then everything is sorted from left to right. So now we're sorting by FOXM1 expression. Now we're sorting by the study. So it gives you a lot of flexibility here. Another thing I want to show you is the chart up here. So the chart is another way to view the data. You can see here, you can always go back to the visual spreadsheet. So in this case, it by default just shows the first column and shows what percentage of samples are in each of these categories. But you can also, I'm going to change the x-axis up here to be study and the y-axis to be FOXM1. I'm going to give people just a moment to get to that. I know it can be a little takes a second for it all to get together. And so you can see here the different, it's just another view of what we saw. So remember the lower expression in the GTEx, higher expression in target and in TCGA. You can go back to visual spreadsheet here. And so everything that we've done so far has been in this really large data set, but I also wanted to show you how this data, you can pull data from more than one hub together. So in this case, we're gonna change the cohort to look at just the TCGA PanCan data. So we're gonna click on here, change cohort. At the top, I'm gonna type in PanCan. I see that I actually have another question. Excellent. Can we, oh, here, let me see, 1 second. So I'm gonna select this one. Can we annotate for the cancer study type along with the source? So yes, let me show you how to do that before I go on to this next piece. So I'm gonna go back, so I'm gonna just click outside. We're gonna stay in this place here, looking at all of this data here together. So I'm gonna click the plus data button on phenotype. In this case, I'm gonna click on category. So each of the, and then this will show, well, here, give it a second to load here. So I think I should note that so there's 3 different large cohorts, 3 different large groups coming together here, TCGA, Target, and GTEx. So for each of these individually, we have the study information. I'm just gonna drag this here because otherwise it doesn't look very great. So you can see here, so we can show, for the GTEx data, we show where the tissue is from. Brain, breast, you can see there's quite a few. For the TCGA, we see what the cancer type is, and then same thing for target. So hopefully that addresses your question. If not, definitely ask it again and give me a little more context. Great, thank you so much. These are really fabulous questions. I'm very excited about them. All right, so now we're gonna go and look at the PanCan data here. So we wanna click on TCGA and pan-cancer here. And that's gonna change the cohort. Again, it always leaves us in the visual spreadsheet view. In this case, we're looking at 12,000, almost 13,000 samples. So we're gonna go to plus data. So we can see here that more data, we're combining data from 2 different hubs here. One from the TCGA hub, which has all the data from TCGA, as you can see, copy number data, methylation data, some data for filtering, gene expression, SNPs. And then it's also combining data with the TOIL data. So here I'm gonna look at gene expression. You can see all the different TOIL data. We're gonna look at the TPM here, and we're gonna do FOXM1, click done. And again, it's pulling a lot of data, so it's thinking just a little bit about this. So you can see here all the different, for every single tumor in TCGA, we can see all the different data here. We can also, similar to how we did for the large group, the TCGA, Target, and GTEx combined, we can look at the tumor type for just TCGA. So we'll click on phenotype. In this case, it's sample type. And then we're gonna click done. And again, it's just taking a little bit because it's looking at 13,000 samples. Oh, I'm sorry, not sample type. Although we are going to need sample type later on, so I'm going to leave that piece of data up there. So it's phenotype. I think it's, oh, I remember, it's cohort. We'll give that just a little second here. Oh, it still has the PanCan data. I will email you, and I forget which particular phenotype column it is. So in any case, I'm going to remove this here. So this is showing actually exactly what one of our questions was, is that, so if we're looking at sample type here, we have some solid tissue normal, and then the primary tumor. And you can see here there is some expression data for the solid tissue normal. But if we want to look at, for instance, if we want to run a Kaplan-Meier plot on this is a really popular use of our tool. Then we want to run it on the primary tumor. We don't want to run it on the solid tissue normal expression. So I'm going to show one of our newest features, which is this highlight feature here. It's a way to highlight samples within the view here, and you can enter data. Anything that's in view, you can search on it. So for instance, here, we want to find samples that are from the primary tumor. So we can type in primary and tumor, and this will find samples that are annotated as primary tumor. There's a lot. I'm just going to show that one little piece, but I just want to show you right here. You can also do a lot more advanced searches than what I'm doing right here. So click on this here to give you some more help with that. So now we have our primary tumor samples highlighted. We're going to click on this. It tells you that of the 12,800 some odd samples that we have in view, 10,500 of them are matched by primary tumor. So we're going to click here, which is the filter button, and that's going to limit the samples that we're going to view to just the primary tumor. And again, because it's so many samples, it takes just a little bit of time. I'm still thinking here. So now we can see it's just the primary tumor, and we can go generate a Kaplan-Meier plot. The Kaplan-Meier plot, because it's running the, it's giving you a p-value as well, actually does take quite, this one probably takes maybe 5 seconds to load, just with this many samples, and then also running the statistics. It just takes a little bit of time here. But I'll, oh yeah. So, okay, I'll just go over this really quickly. So yeah, so you can see here that if you have lower expression of the FOXM1, you tend to have better survival compared to the medium and the high, and it is significant here. So, and then the last, oh yeah, so my coworker here reminded me, so to get the, that tells you the type of cancer that it is, it's primary disease. So see here, primary disease. You click and then it'll add that. Again, taking just a little bit of time. While that's happening, I'm going to go and see, oh, here we can see. So I'm going to move this up here so you can see all the different cancer types here. Excellent. Okay, we have some more really good questions here. Okay. Okay, let me see which one would be best to answer first. Okay, so I'm gonna do this one first. The question is, how can we identify TCGA version number being used in the UCSC Xena browser? So this is a really great question because you always want to make sure you can reproduce your data, right? So in this case, on the caret menu and click on About Dataset. This will open a new tab that'll look at that particular dataset, and it has the version here. So this is the version of last time we downloaded the data from TCGA. So that's a really great, so then you can say, I use this version of the data. Excellent. I'm going to answer this one next, and I'm going to ask, so the question is, would it be possible to repeat survival curve plots? In this case, I'm actually not quite sure what you're asking. So you can, at any point in time, you can always rerun the same KM plot. So we just did a KM plot here. We can rerun it again. In this case, it's in memory, so it pulled up really quickly. But yeah, so we just repeated it here. So I'm not quite sure if that's what you're asking. Maybe if you could reword it, that would be really helpful for me. Thank you. So now I'm gonna select this one. What is the difference between the PanCan TCGA and the PanCan target, or the PanCan TCGA target, or the PanCan TCGA target GTEx? Are they not subsets? Yes, so they are subsets. So we have just put together, so for TCGA target GTEx data, we just put them all together in the same group so that you could view them all together. Same thing for the PanCan TCGA and target. We just put TCGA and target data together in the same group, so it's easy to view them together. So they're all, so the TCGA data in this case, the TCGA data in the PanCan TCGA and the PanCan TCGA target and the PanCan TCGA target GTEx, that data, the TCGA data, is exactly the same across all three of those groups. Target is the same across those two groups, GTEx is the same. So, so yeah, so they're just, we created these as a way for, because some people kept on asking us, oh, I really want to be able to see the TCGA data and the target data together. And while you can combine them together, we have an application to be able to combine them on the fly. We just had so many people asking for it, we just created it by default so that it was a little bit easier for people. So yeah, they are subsets. Excellent. And then I'm just gonna answer this one here. It says, how can we download bulk UCSC raw/normalized data sets? And so I answered this in the beginning, but I wanted to put it here for other viewers as well. So we have an S3 button, and you can bulk download it there. So I'll send out at the end of this webinar a link to that S3 bucket to everybody who is here, so that way anybody can access that. And then, so I think that's done. Are there any more questions? And I just want to, hopefully I answered that KM plot question correctly. I still want to make sure that I'm correctly understanding and answering what you're asking. That's important to me. So at this point, this is the end of the example, all the examples that we had. It's 11:45, so I'm gonna wait just 5 more minutes, see if anybody has any further questions. So far we've gotten a lot of really great ones, which is wonderful. And if anybody has anything else, feel free to email. Excellent. So here we go. I'm going to select this. So how frequently is UCSC Xena datasets, or how frequently are they updated for TCGA, GTEx, and target? So during the heyday of TCGA, when there was a lot of updates, updates happening. We tried to update it maybe once a month just because TCGA itself was releasing so much data. Right now, TCGA is, it's almost finished. There are very few data is coming, basically completely done with their analysis. So at this point in time, we're updating about quarterly. And that seems to get, every time we update it, we get maybe one more sample for, breast cancer, that sort of thing. It's very, very small, the changes that are happening. For the GTEx data, we actually just got that data set in the last couple of months. And so it's relatively new. I imagine that right now, so GTEx is still in their final stages. They are updating frequently, but they're having releases. So for the next time GTEx releases data, which I believe is going to be in maybe 3 or 4 months, we'll update it when they tell us, okay, we have the newest data. So we'll make sure that we keep up to date with their releases. And then the target data is not being updated very frequently right now. So we check in with them, but it's very similar to GTEx as they're updating, we update. So great question. And then yeah, so always make sure to pay attention to that version number by clicking the About data set. Click Done here. Just so you know, you can also click on, well, here. So we're in the Explore data here. So this gives you more information about, you can download the data, but you can also see what other data sets are available. This is all in the Explore data. So Explore data is basically looking at all the different data we have and sort of giving you an overview of what kind of data we have. You can also see the different data connecting to here. This is important if you start connecting to your own personal hub. Oh, excellent. Okay. Got it. Okay, I'm just making sure I covered the survival plot. Perfect. Excellent. So glad that this was helpful for you. All right. So again, I'm just going to wait maybe 2 more minutes for anybody who is typing out something to ask anything. But I think that's it. So thank you, everybody. And we'll probably, right now we're tentatively thinking of doing this. We'll definitely be doing this again. And then we're just sort of deciding on how frequently we want to do this. So maybe not always for the TOIL data, we might do a more general one as well. So excellent. I guess the one thing I will show, just so it's here, is that if you want to download your own Xena hub and load your own data into it, you would click here in the local data, or in the local Xena. So that will give you, you can just click on this big button here, download platform. So that gets you set up there. Excellent. Okay, I think that's it. Thank you, everybody, and we'll feel free to write into the mailing list or send an email to me if you have any follow-up questions. Bye-bye.